very happy to share with you some results of our work on human genetic diversity and metabolic diseases in North Africa. Okay. The plan of my presentation is the following. So I will start with an introduction on prehistoric and historic background of North Africa. Then I will present genetic diversity of current population and prehistoric North African populations. And finally, I will present the role and the impact of genetic variability of North Africa on metabolic diseases. So let's start. As you can see on this map, North Africa is located at the crossroad of Europe, the Middle East, and Sub-Saharan Africa. Thanks to this geographic position, North Africa has known multiple migration waves and invasions. So I will take the example of Tunisia to show you some details of prehistoric and historic events. And that are the same, closely the same for other North African populations, in particular, the western north of Africa. So Tunisia was occupied by the Etruscan civilization during the Paleolithic periods. And in the Epipaleolithic periods, the Ibero-Mauritian civilization succeeded to the Aterian. You can see here in this map that Epipaleolithic civilizations occupied the littoral of North Africa. And the main archaeological sites are Tafouralt in Morocco, Kolumnata, and Afalou in Algeria. In the Mesolithic period, the Capsian civilization appeared in central and southern Tunisia for about 8,000 years ago. And Capsian man is considered as Proto-Mediterranean. Let's move now to the historic period. Berbers, called also Imazic, belong to the most ancient group, which seem to have Capsian as local ancestors. Along its 3,000 years of history, Tunisia was successively under the reign of Phoenicians, Romans, Vandals, and Byzantines. Since the second half of the 7th century, date of the Arab conquest, Tunisia became Arabic Muslim. The 11th century was marked by the invasion of Bedouin, originated from the southwest Arabia. And it's considered as the second large Arab immigration into Tunisia and accelerated the process of Arabization. The 15th, 16th, and 17th centuries were characterized by the mass migration of Moorish Andalusian Muslims and Jews expelled from Spain to North Africa. And finally, during the 19th century, French forces established protectorate in Tunisia, and French occupation ended in 1956. So, as you saw, North Africa has been occupied by several civilizations at both prehistoric and historic periods. Our question was how these invasions has shaped the genetic landscape of North African populations. To answer this question, we proposed to study mitochondrial DNA diversity and to perform phylogenetic analysis. So, firstly, allow me to explain why we choose mitochondrial DNA. Mitochondrial DNA is located in mitochondria, and it's considered as an important tool for genetic populations, because many features, as the small size of this circular DNA, maternal inheritance, absence of recombination, and high mutation rate compared to nuclear DNA. So when we study mitochondrial DNA sequences, we do alignment of these obtained sequences with the reference, and this allows the identification of polymorphism. You can see here some polymorphism and haplotype. And then this haplotype is classified in haplogroup. And the majority of mitochondrial DNA haplogroups are specific to continents. So here, for example, you can see all haplogroup L from LO to L3 or others, L6, L7, are originated from North Africa, for example. So consequently, mitochondrial DNA is a powerful tool to study the settlement of regions and to draw migratory waves. For our study, we sequenced mitochondrial DNA from more than 150 individuals from northern Tunisia and from other regions here located in red stars. And also, we collected more than 765 mitochondrial DNA sequences from 16 Tunisian populations from literature. And these populations include 3 communities: Andalusian communities, Berbers communities, and Arab and people, Tunisian people with Arab origins. In addition, we collected mitochondrial DNA sequences from North African population, Near East, and European population. We used more than 5,000 sequences to perform our phylogenetic analysis. Here are our results. So our findings showed that the genetic structure of Tunisia is a mosaic highlighted by the presence of a wide variety of haplogroups, 22 haplogroups classified in 3 components: Eurasian components, Sub-Saharan lineages, and North African lineages, with the predominance of Eurasian lineages. So the high variability of Tunisian population is also showed even between localities very close and between population with the same ethnic background. I give you an example here of 3 Berbers populations in the south of Tunisia belonging to the same department in Gafsa, and you can see the high level of variability between these 3 communities. Another example, these 2 Andalusian populations, very close geographically, they are only 5 kilometers between the 2 localities, and you can see also the high level of variability and the difference in the distribution of these haplogroups. And finally, capital Tunis here show the highest level of variability in Tunisia, but also at the Mediterranean region. And we can see after in the next slide this heterogeneity. So the high level of heterogeneity is not only observed at the level of Tunisia, but also at North Africa as well. So here you can find several North African population, Algerian, Moroccan, and Libya, and they are the 3, we can observe the 3 lineages: North African lineage in green, Sub-Saharan lineage in red, and Eurasian lineage in blue. And here they are high level of variability compared to Near East population and European population. So our phylogenetic analysis performed with more than 5,000 mitochondrial DNA sequences from Mediterranean populations, including, of course, North African. Here you can see that this cluster is composed by North African population. And you can see there, this cluster is heterogeneous compared to the cluster of European or Near Eastern. So here we have also some North African populations clustered with European populations and others very close to Near Eastern. So we can conclude that North African population are more heterogeneous compared to European and Near Eastern populations. It is important to note also that there are 3 populations in North Africa, 2 Berbers from South Tunisia and Berber from North Algeria. Here we are very genetically distant from the other North African population and can be considered as outlier. And not only at the scale of North Africa, but also at all Mediterranean or all the Mediterranean region. So all these observations confirm the high level of variability of North African population. For more details on this work, I invite you to see our work on phylogeny and genetic structure of Tunisia and their position within Mediterranean populations. So let's move now to the genetic diversity of prehistoric populations. So to better understand the origin of North African settlement, we studied the mitochondrial DNA diversity of Ibero-Mauritian populations. So we studied skeletons from the archaeological sites of Taforalt in Morocco, dated from 23,000 years before present to 10,000 years before present, and the archaeological site of Afalou in Algeria. So here, the cave of Taforalt, located at 55 kilometers in the northwest of Oujda in Morocco, we studied 30 specimens. And here, the archaeological site of Afalou in Algeria, near to Bjeya. And we have available only 7 individuals from this ancient collection. So DNA was studied and was extracted from bone, and we amplified mitochondrial DNA with real-time PCR, and the DNA was directly sequenced. So this is our results. The genetic structure of Taforalt population is composed by 2 components, the Eurasiatic component and the North African component. And so we can see the absence of the Sub-Saharan African component observed in the current population. It is also important to note that GT is a very rare haplogroup currently. It's only observed in Berbers from northern Morocco, in Sicilian and in other Italian populations. For the population of Afalou, the genetic structure is composed mainly by Eurasiatic components. So we note the absence of Sub-Saharan haplogroup. Here, our phylogenetic analysis showing that Taforalt and Afalou are clustered with North African population, mainly with Berber of North Africa, and with European population from Southwest Europe. So we can conclude here that from this analysis of prehistoric population of Afalou and Afalou, that they are a genetic flow between southern and northern coast of Mediterranean, and there is genetic continuity in North Africa since the Epipaleolithic period. Our results can help to precise the chronology of migration and the settlement of North Africa, mainly the Sub-Saharan African lineage, which appeared in North Africa after 10,000 years before present. To have more details on this work, I invite you to see our work on phylogeny and genetic structure of the, sorry, it's not this. This is, no, it's a mistake. Okay, I will give you the title of the work on Ibero-Mauritian origin with through mitochondrial DNA analysis and phylogenetic analysis. Okay, the take-home message from these 2 studies is that there are a high genetic heterogeneity of North African population compared to European and other Mediterranean regions, genetic continuity in North Africa, and the existence of genetic flow between southern and northern coast of Mediterranean since Epipaleolithic period. And we precise the chronology of Sub-Saharan lineage. This Sub-Saharan lineage observed in the current North African population is due to gene flow from South to North Africa after 10,000 years before present. So our finding is also confirmed with other work performed on another uniparental marker, which is Y chromosome. So several studies performed on North African population showed that they are also a mosaic aspect of this genetic structure of North Africa, considering Y chromosome variability. We can have Eurasian lineage, Sub-Saharan lineage, and North African lineage. And what about other nuclear markers are with Mendelian inheritance? Here I can answer to this question by this work performed in our lab during the PhD of our colleague Yusra Ben Halima. And this work is on lactase persistence in Tunisia. So when we studied the genetic region involved in the lactase persistence, we observed, this is an admixture analysis, we observed that North African population, including Tunisia, the genetic structure is mosaic with the presence of an African component, European component, Asiatic component, and Sub-Saharan African component. So our finding with the mitochondrial DNA are also confirmed with other markers, uniparental marker like Y chromosome, but also other markers with Mendelian inheritance. Another particular aspect of North African countries is the high level of consanguineous marriages. So our recent studies showed that the rate of consanguinity in Tunisia is around 30% in northern Tunisia and can reach over 60% in some areas, in particular in the south of the country. We showed also that the rate of consanguinity is around 20% to 50% in Algeria. So this consanguinity impact on the genetic diversity of population. And I show you here one work on an isolated Berber population from South Tunisia. This work was performed during the PhD of our colleague Yusra Ben Halima. And here, this isolated Berber population presents a very particular genetic structure highlighted by the high level of the T haplogroup, which is an Eurasian haplogroup. Our phylogenetic analysis showed that Duiret, this is the isolated Berber region from the south of Tunisia, is genetically very distant from the other North African population and Mediterranean populations. To see more details on this work, I invite you to see the paper on mitochondrial DNA structure of an isolated Tunisian Berber population and its relationship with Mediterranean populations. So let's move now to the third part of our work regarding the metabolic diseases and how genetic variability can impact these metabolic diseases. So you know now that there is an epidemiological transition worldwide characterized by an increase in communicable diseases, multifactorial metabolic diseases mainly, due to the change of lifestyle. So according to the International Federation of Diabetes, more than 463 million of persons are affected by diabetes. In North Africa, type 2 diabetes is around, the prevalence of this disease is around 10 to 18%. But what is more alarming is the metabolic syndrome. The prevalence is between 13 to 30%. So metabolic syndrome is a very complex metabolic disease due to the presence in the same person of several metabolic abnormalities. So we can find in the same person more than 3 or 5 abnormalities, including central obesity, hyperglycemia, hypertension, insulin resistance, and dyslipidemia. So this syndrome at long term leads to complications and death of patients. And also this, you can see here in this figure that these complications can affect several organs and several tissues. And according to the International Diabetes Federation, metabolic syndrome, which is leading to diabetes, for example, that there are more than 4 million of persons of deaths every year due to diabetes, which is equivalent to 1 death every 8 seconds. So to show you how the variability and the genetic variability can impact the metabolic diseases, I take here an example from our work on a pharmacogenetic landscape of metabolic syndrome drug response. This work was performed during the PhD of Ms. Haifa Jmal. And this is the first study of pharmacogenetic landscape of metabolic syndrome, not only in Tunisia, but also in North Africa. So now it is well established that pharmacogenetic profile, it is very important because they are inter-individual variability in response to drug. So it depends on the pharmacogenetic profile. Patients can present high risk of adverse drug reaction, moderate risk, or low risk of adverse drug reaction. Until now, there are no specific medication for metabolic syndrome. The therapeutic strategy consists on an individual management of each component of this syndrome. So, for example, patients are treated with oral antidiabetics in order to treat hyperglycemia, or anticoagulants for other vascular diseases problems. So the aim of our work is to characterize the genetic variability of pharmacogenes involved in the response to metabolic syndrome treatment. To perform this work, we selected very important pharmacogenes involved in the response to treatment, to medication, usually used in the treatment of metabolic syndrome patients. For example, anticoagulants, oral antidiabetics, and lipid-lowering agents. So then we studied the variability of these genes, and we compared this variability with 20 populations from the literature and from public data. Population included North African, population, Asian, European, and American population. So here our results. The multidimensional scaling plot analysis displays 3 clusters. You can see here this cluster is grouping, consisting with North African populations with clustered with European population. So you have Tunisian, Algerian, Moroccan, Egyptian here with other European populations. And this cluster is distinct from sub-Saharan African populations and the Asian populations. The analysis of the admixture is used to infer ancestral populations group. So here you can see that North African population here is a mosaic with the presence of North African, European, and Asian components. So the genetic structure is regarding to this very important pharmacogene involved in the treatment and the response of to metabolic syndrome are influenced by the genetic variability, and are influenced also by the different gene flows that now appears in North Africa since the prehistoric time. So we confirm also our finding of mitochondrial DNA or other genetic markers. So in summary, we highlighted the high genetic heterogeneity of North African populations compared to European or other Mediterranean populations. It is important to take into account this specific genetic structure in the design of future association study and clinical trials. And it is important to identify the pharmacogenetic profile in order to optimize drug dose and to avoid serious adverse drug. I finish with this Roman mosaic, which reflects the mosaic structure of North African population. Here are my 2 email addresses, so you can contact me if you need more details or more paper. Thank you very much.